Hello, everyone. Welcome to the Life Science Rundown. This is Nicholas Capman with the FDA Group. Before we jump into our discussion for today, just a little bit about the FDA Group. We help life science companies in the areas of quality assurance, regulatory affairs, clinical operations, commissioning, qualification, and validation, CMC, as well as pharmacovigilance. We offer 3 different engagement models, which are consulting, staff augmentation, and full-time employee recruitment. So if you ever find yourself in need, just head over to the fdagroup.com to check us out and get in touch. So today I'm speaking with one of our consultants, Gary Ritchie. Hey, Gary, how are you doing today?
Good morning. It's good to
Okay,
see you again, Nick.
absolutely. Thank you for joining me. So before we jump into our topic for today, would you introduce yourself?
Sure. I'm a pharmaceutical scientist. I've been practicing for almost 30 years. I have about 20 years of bench experience. I had the interesting experience of moving on to the United States Pharmacopoeia, where I served for 5 years on the expert committees for water, statistics, and general chapters. I was the liaison, which allowed me to work with the FDA, industry, and academia. Once I established expertise in some of these areas, I had the opportunity to go off and work for multiple companies after that. And so I got into consulting, taking some of my experience in statistics, some of the instrumental methods in analytical chemistry, as well as a little bit of experience in pharmaceutical waters. It's expanded much more beyond that now. But I've been to make a good run at it since then, and look forward to continuing to work with groups like yourself and others.
Okay, excellent. Thank you.
Thanks.
So Gary, what are we going to be talking about today?
So, one of the areas that I had a chance to think a little bit about when asked, what's your opinion of the state of uh, sampling today, sampling in general, sampling from receiving shipments to sampling in the lab. I got into process analytics some time ago, and there the issue of sampling comes up. And one of the standards that have been used and relied upon for some time is based on the square root of n plus one approach. So, having thought about this and working with some clients, we're going to be delving into the idea of statistical thinking and acceptance sampling for biopharmaceutical raw materials, and the best practices that are used today, and where I think they should rely on more formalized statistical testing.
Okay, excellent. What makes a statistical sampling plan valid?
So we will get into the idea of just exactly first what square root of n plus one is. And it's generally been used to apply for large lot sizes. It has a history from the United States Department of Agriculture, where they were looking at sampling from fields, right? Very large population. It was very low cost, and it typically led to some destructive testing. But in general, the requirements are not met, and that's what brings us into the application of biopharmaceuticals. The requirements in the 21 CFR 820-250 for statistical requirements are not met with the square root of n plus one. And so that's why we're going to discuss it in detail and find out why and where it leads us to better approaches.
Okay, excellent. Why is the square root of n plus one rule not useful for small lot sizes? And what is a small lot size statistically?
So, if approaching the use of square root of n plus one for small lot sizes, which is what we generally run into with biopharmaceutical samples, typically from 5 to 30 samples, right? It's not valid. And so the question is what makes a statistical sample valid in the first place? Well, number one, the rule of thumb is any draw from a population must be representative. That sample has to be representative, which means that any sample that is drawn from that population should have the same ability to be representative of the whole lot. Okay, so you can understand that that frequency of getting a representative sample that is descriptive and has the properties of that whole lot statistically will decrease as that population size increases.
Right.
So how do you deal with that? First of all, the lot size has to be accounted for, right? With what we call the ANSI Z14 attribute sampling, the next thing that allows it to do that you don't have control over with square root of n+1 is the inspection level, right? Which really takes care of the risk of drawing the wrong sample. The smaller lot sizes will generally have a defined inspection level, and it will be lower and less than an inspection level that's for larger lot sizes. Okay, this ensures, right, that you are getting a better representative sample. And the third thing is the acceptance quality level, which is called the AQL. Okay, those three things plus representativity is what makes a valid sample plan.
Excellent. So what is the solution?
So because we have generally small samples, as I said, for biopharma samples, between 5 and 30, if not even less, the true data of the population value has to be assessed properly, which means that you have to have the right inspection level. You have to be able to select the inspection level according to a lot size. And out of those two parameters, you end up with selecting an appropriate AQL. And geez, if we had a table, I can show you exactly how that works. We have some great references that I relied on to understand this a little bit better and hopefully to be able to explain it better. But once the rule is applied correctly to different sample sizes, you can begin to actually observe the effect of sample size on what's called the AQL curves. And these curves are generally what are used to assess the lowest and the upper range in which is the most appropriate sample to that'll be representative for the selection. So it turns out then most people will say, well, then why do we use the square root of n+1? Because mathematically, it's almost that you can't differentiate a square root of n+1 from an AQL curve. They pretty much match in a narrow range of sample of lot sizes. And as it turns out, the larger the lot size, the more approximate that square root of n+1 gets. The smaller the lot size is where we run into the problem. And that's why I thought it was very much applicable to biopharmaceutical sample, where, for instance, where you're looking at tissue selection, reagents to grow mammalian cells, for instance, okay? Sometimes a pharmaceutical company will only get 1 or 2 lots a year for that. And so the question is, you know, what do? Test them both? Well, if it's destructive testing, you can't. So can you get away with just one? Yes. But then what is the risk of that? You can't answer that question with square root of n+1. Okay. So that's the first issue, is the square root of n+1 doesn't accommodate the risk factor. The second thing I think that you have to deal with is you may run the risk as you use the wrong sample size for smaller lots. Generally, you run into the problem of oversampling. Well, and if you're oversampling, then your estimate's going to be wrong. Your risk approximation may be correct, but you still could fail the whole the whole approach. So in a nutshell, one has to be cognizant of managing the risk and what's the appropriate sample size based on the lot size that you have.
Yeah, makes sense. So what does this all mean?
So I think you can sum it up by saying this. If you take into effect the lot size and the risk factor, what does it mean? Well, in a business, ultimately it impacts the bottom line. And that means that it's very costly to continue to use the square root of n+1 when essentially we have a way to minimize the cost to the whole thing. And number two, the risk to the patient is managed much better because you have a lot more information based on the correct sampling size and based on your ability to assess that risk and manage that risk. So it comes down to the impact to the patient and the impact to the business. That's the two places that we get the advantage of using the correct statistical sampling. So I do recommend, and we're talking about attribute sampling in general. The appropriate message here then is one for businesses to understand that, the FDA has accepted and used this now for a long time. And the issue there is, well, if that's the best practice and it's seems to be compliant with the FDA's wishes, there shouldn't be any impact. There's no pressure, right, to move off of the use of square root of n+1 and move into the ANSI sampling, either for attributes or, and I started to say earlier, there's another ANSI mechanism for continuous sampling where you have not the property of the sample that you're testing for, but you have a range of values. Okay. This could be looked at probably in, let's say, continuous online batch manufacturing. However, you aren't compliant with the FDA. Now, whether they accept it or not, that's fine. But we went over the rule, right, in 21 CFR 820 for statistical sampling, and you're simply not compliant. Do you leave yourself open to the FDA scrutiny of your sampling, acceptance sampling plans? Yes, you do. So that's, in a nutshell, the three issues that we're dealing with, right?
Yeah.
Cost, risk to the patient, and compliance.
Yeah. Well, this is really good information. It was very thorough yet concise. Before we wrap up the podcast, what would you like to leave the audience with?
So, I had to delve into this, and the first thing that people want to know is, well, where do you go to learn about the approach? Well, you can go to the ANSI standard, which is available online, I think, for purchase. The approach that I took was to do literature search. Anything that people do for research is the best place to start is what's been written about it. And so learning ANSI has been the barrier to adoption. That's for sure. We know that. As soon as you say the word statistics, I've seen all the way from CEOs down to, lab analysts at the bench roll their eyes and go, oh, geez, I know how to do standard deviation, I know how to do mean, right. But I don't know how to implement a complicated. Well, number one, I'm going to demystify the idea that ANSI is complicated. It is not. It's just that you don't know how to use the table. So you learn, you teach yourself, and then you begin to understand how it works. The more samples, the more different types of lots and methods that you have to come up with to draw samples from, and you generate those AQL curves, you can actually begin to, in your mind, draw the picture of what the statistics is doing as you increase or decrease the lot size. Yeah. But once you see that, you have that feeling that, hey, I know what this is. And that's the key. So who does that better than anybody I have seen write about it was Dan O'Leary, wrote a paper called Attributes, Acceptance Sampling, Understanding How It Works. I have that reference. That paper was, I think, instrumental for me. That was written in 2009. He was with OMBU Enterprises. And then the second paper that I thought was instrumental in helping me understand the regulatory impact of using ANSI was Ron Snee, a well-known lecturer and expert in this area. Solving statistical mysteries. What does the FDA want? Well, the answer to the question of what does the FDA want, we all know what that is. They want you to comply. And regardless of what square root of n plus 1 has meaning to the industry and to the regulatory agencies around the world, not just the FDA, again, it's the penguins going off the cliff. Just because one jumps off, do all go off the cliff? And is that the right move to make? That's what we're talking about today. It may be the most used approach. Is it the right one? I think as we move more and more to smaller lot sizes, as we start to see more and more problems in the biopharmaceutical area, and we can see how that's going with the FDA for increased 483s, warning letters as industry moves in that area, will begin to understand, and they think the FDA will begin to understand that they have to get a rope around this problem. And I think this is a solution if industry wants to get ahead of them.
Yeah. Awesome. So, I think this is all excellent information. I really appreciate.
That's a quick and dirty,
Yeah.
I don't know if there's a lot more to it, obviously, Nicholas, the surface we're scratching here. But I think people just have to get in there and get their hands dirty. I did. I was a non-believer. I was like, well, it works. Why do I want to go over here? But you can see the 25 to 50% savings. What CEO or what business unit would not grasp at that?
Right.
So that's the issue.
Yeah. Well, even though we scratched the surface, I still think this was really helpful. And I'm sure, the audience got something out of it. So I really want to thank you for taking the time and sharing your insight on this topic. And, for everybody listening, as always, if you find conversations like this valuable, please share it with a colleague or your professional network. If you haven't already, subscribe to get updates of new episodes at thefdagroup.com/podcast and follow the FDA Group on LinkedIn. If you'd like to connect about project or resource needs, head over to thefdagroup.com to get in touch, or feel free to connect with me personally on LinkedIn. Thank you so much for tuning in, and we'll see y'all next time.
Thank you, Nicholas. Thank you, audience.